हे गाइस, क्या हालचाल। नया दिन नई शुरुआत। ड्राई लैब वर्सेस वेट लैब। जी हाँ, हमेशा हम अपने आप को एक पर्टिकुलर कैटेगरी में फिट नहीं कर पाते। हमें पता ही नहीं चलता दैट डू वी बिलोंग टू वेट लैब और डू वी बिलोंग टू ड्राई लैब। वेट लैब मतलब कि केमिस्ट्री, सिंथेसिस या फिर फार्माकोलॉजिकल एक्सपेरिमेंट्स या फिर फार्मास्यूटिकल फॉर्मुलेशंस, एनीथिंग व्हिच डील्स विद instruments, devices, okay, softwares, chemicals, anything that deals with that is a wet lab. Microscopy, staining, histopathology, anything. On the other hand, dry lab is basically sitting using a particular device, using a particular interfaces, profiling, lot of different things, right? In silico, basically. All the work that is in silico, that you don't have to use the chemicals, that is called as a dry lab. Now, when I talk about an overview, so in this video, we are going to discuss about the dry lab, wet lab, the difference between the dry and the wet lab and their applications. So a wet lab is something where you have hands-on experience. You perform the experiments, you use biological samples, chemicals, and like example, microbial culture. So right, you need a culture media, you need an inoculum, you need to culture the particular plate, right, a particular agar medium. Drug testing, for testing you need to inject the drug in a particular vehicle or and then into the animal, correct? You need to dissolve a drug. Extraction. Dry lab, you do a computational work, a simulation based work, which focuses more on the data analysis, modeling, virtual testing, etc. So it uses all databases, softwares, no physical experimentation. And both of them, have their own advantages. Okay, it's not that wet lab is better or dry lab. Both are interdisciplinary, both needs each other. Correct? In silico proceeds is proceeded after the in vivo in vitro, right? So yeah, this involves bioinformatics, statistical analysis, etc. So these are the key differences. In the wet lab, there is experimental hands-on experience. In the dry lab, computational work, analytical techniques. Wet lab, there is benches, fume hoods, pipettes, biological samples, chemicals. Dry lab, software, tools, databases. The risk is a little high in the wet lab, which is because you deal with a lot of chemicals. They could be carcinogenic, teratogenic, mutagenic, genotoxic, anything. They might cause mutation. So these are some of the major differences between a dry lab and a wet lab. Types of wet lab: molecular, microbiological, pharmacological, chemistry, pharmaceutics, analytical, tissue, right, formulation. Dry labs types: biostatistics, machine learning, epidemiological studies, PKPD modeling, molecular docking, chemoinformatics, bioinformatics. So the application of wet labs are in the drug discovery and testing, vaccine development, toxicological studies, clinical sample testing, while the applications of dry labs is in the predictive drug modeling, genomic data analysis, clinical trials, AI-driven biomarkers. Like there are a lot of AI-driven software nowadays. AI is doing everything. So dry labs are gaining a lot of populance nowadays. So this is a basic difference between a dry lab and a wet lab. I hope that this was very useful for you. You would have got to learn a lot of things for sure out of the dry lab and the wet lab. So for this video, that's it. Thank you so much for joining for today's video. We'll be sharing up more such informative videos in the upcoming lectures. Thank you so much.